The domains bacteria and archaea are two of the prokaryotic groups that we have looked at in class. And when we remember, prokaryotic cells simply are those cells that do not have a nucleus, so that their DNA is going to be contained within a nucleoid region, nor do they have membrane-bound organelles. And those membrane-bound organelles are going to be those specialized compartments that are able to transport material, make the energy, things such as the mitochondria, the endoplasmic reticulum, the Golgi apparatus. Those are all going to be specialized organelles that are found in eukaryotic cells. So the prokaryotic cells, bacteria and archaea, do not have a nucleus, nor do they have membrane-bound organelles. What they do have, though, are what all other living organisms will have. They will have those characteristics of life. They are able to reproduce. They are able to get and use energy. They are able to use energy. They're able to adapt. They're able to evolve. They're able to respond to stimuli. So all of these different characteristics of life, which are all in Chapter 1, Section 1, all of those characteristics of life, these two groups, these two domains do indeed have. When we look at their size comparison, most of us can kind of distinguish a period out of a sentence, maybe a little bit smaller. The bacteria that we are going to be trying to hopefully observe with our microscopes are quite small. They are going to be on a scale of micrometers. Mainly, a lot of them are about 10 micrometers at most. Viruses are even smaller. Viruses can actually even infect bacterial cells. Viruses can also infect our eukaryotic cells. So this is kind of giving an idea of the scale with respect to from water to glucose. Those are two things that were able to pass through that plasma membrane that we had in class, to a little bit larger, to ultimately the size of a tennis ball. So you can kind of get an idea, hopefully, of the perspective of how small bacteria are. When we look at the microbiology, we're looking really at small things. If I were to extend this centimeter ruler, that would be approximately 1 inch, which many of you are familiar with. It's probably 2.64 centimeters is going to be an inch. What we're going to be looking at is in this region here. This is 1 millimeter. And 1 millimeter is the same as, in terms of unit value, of 1,000 micrometers. So we're thinking about, say, 10 bacteria, or if bacteria are approximately 10 micrometers, 10 bacteria can fit in this little bitty region of a ruler. And if we got to even a smaller scale in terms of the nano, we're looking at a much smaller group. That area is going to be approximately 1 million nanometers. So this is the region that we're going to be observing with the microscopic world that we're about to enter into in our study. Many microbes are going to be beneficial. Not all of them are going to be harmful. As a matter of fact, really the only harmful ones are those which are considered to be pathogens. And pathogens are those harmful viruses, bacteria, or fungi, even protists that can cause humans illness. Many of them are going to be much more favorable in terms of decomposers. Think about all of the material that Slafter takes from the cafeteria. If you've never followed him out, go and watch him put that compostable material in the compost bin and come back a few weeks later, you would see a bunch of bacteria. Well, not necessarily see them, but smell them. You might be able to see the fungi. These are the microbes that are breaking down all of the compostable material that we are producing. Some of these microbes are also photosynthetic. Cyanobacteria are also considered blue-green, and they were miscategorized about 30 years ago. Scientists thought that they were algae, and so you might see something such as cyanobacteria algae or cyanoalgae. It's now for sure known that Oscillatoria, nostoc, these photosynthetic bacteria are going to have this blue coloration. Some of them are called blue-green, and the pigments, regardless, what will happen is they will all have chlorophyll. So chlorophyll A and usually chlorophyll B, these are the protein pigments that are able to capture the sun's energy. Some protists, such as the green algae, such as Chlamydomonas, Spirogyra, are also going to have chlorophyll A and B. They will differ in light of, for example, the cell wall, the cell capsules of bacteria will have a protein, whereas many of the cell walls of this photosynthetic green algae are going to be very similar to plants in terms of storing it as cellulose. The microbial world is also pretty big in terms of biotechnology. Here and around the Bay Area, we have Genentech as one of the main super biotechnologies, and this is where we're looking at how bacteria transform themselves, how some bacteria have actually been produced so that they are oil eaters. And one of the times I remember a few years ago, one of the spills in the Gulf, scientists can go ahead and distribute these oil eaters into the water. And what they will do is the bacteria will consume all the oil that has been spilt and then die off. They will crash because there is no more food left for them. And the bacteria are going to be categorized according to their shape predominantly. We have the rod-shaped, or the bacillus. The spherical shaped are the coccyx, spherical, and then the spiral shape, which is often similar to a corkscrew. These are going to be, the corkscrew bacteria, or the spirillum, are usually going to be single-celled. What's unique is that it's often thought that these single-celled spirillum may well have led to the eukaryotic organelle called the flagellum. The rod-shaped bacteria, they can cluster. They can make two of them, same as spherical. So you sometimes might hear something like diplococcus or say streptococcus, maybe you have a strep throat. That's when these spherical bacteria really join in terms of long chains. So the bacteria, although they're all single-celled, they can join in terms of being diplo. You see some adjectives in here. If they grow in groups, that's usually what they mean staph. Or you might hear staph infection. That's really a type of bacteria that is going to cause an infection such as ringworm. So they're all single-celled. They all also can join, with the exception of the corkscrew bacteria, the spirillum. They're often going to be just single-celled. Classic in terms of your textbook with respect to the structure. Here we have this DNA. All bacteria will have a DNA, and it's going to reside often just prior to dividing in a circle. So you might see a circular DNA in light of the bacteria that it might be ready to undergo binary fission. All bacteria will also have ribosomes. So they will also have the same function, cytoprotein synthesis. So ribosomes, regardless of whether they're for bacteria or whether they're going to be eukaryotic cells, will have the same job of synthesizing proteins. Many of them will also have this capsule, which can be kind of sticky, and the capsule is going to have a particular protein, a peptoglycid. I'll go look it up for you. And the capsule, being sticky, it can therefore adhere to different environments. They might have some storage granule, but notice it's a granule. It's not going to be a vesicle. And often they might have these pili or fimbriae. These pili, which is another word for them, they may well indeed play a role in light of a type of reproduction called conjugation, which is a way in which some bacteria can get new DNA. Okay, so unlike some of you might have heard in terms of asexual reproduction in the form of mitosis, bacterial reproduction is simply called binary fission. The idea is it takes one bacteria, and approximately every 20 minutes this one bacteria can divide and make two bacteria. So really, when we have one single bacterium that might survive, let's say, an antibiotic treatment, if that entire antibiotic treatment is not taken, it's very possible that that bacteria could become resistant and then continue to divide. And after 40 minutes, instead of one bacteria, you now have four, and each of those subsequently continue to undergo binary fission. There are other ways that the bacteria can get new DNA or genetic variation. Conjugation. Often you might see a conjugation tube. Two bacteria are literally side by side to each other. The plus bacteria will donate a plasmid to the negative bacteria. thus exchanging genetic information. Transformation is when small pieces of circular DNA, called plasmids, can often be picked up by the bacteria in the environment. Next semester, you'll actually perform a transformation experiment. Transduction is often when viruses are going to be responsible for providing bacteria with new DNA that the viruses have taken up from other living cells. And like I said, binary fission is quite simple. The DNA will loop itself into two distinct loops after it's copying itself. So here's the original DNA, and here you see the two, I'm going to say circular DNA, as they're beginning to replicate as DNA. Once the new DNA is replicated, each strand or each loop of DNA will go to the extreme end of the bacteria, and eventually the cell membrane and subsequently the cell wall and the capsule will divide, and now we have two bacteria which are identical, or they are indeed clones. The idea is many bacteria are called and considered to be strains. You might have a strain of bacteria that is going to be a heterotrophic, chemotrophic, or photosynthetic. These are essentially how they're going to get their energy. So a heterotrophic bacteria must consume, and often bacteria can slide around some slime. The slime can pick up an extracellular digestion. Can occur because these bacteria do not have mouths. So subsequently they release enzymes to the outside, and these enzymes will break down the organic matter and then small enough so that the bacteria can then absorb it, usually by active transport through the plasma membrane. The other groups are the chemiosynthetics and the photosynthetic. Obviously, from the light, the chemiosynthetic can make their own food, but from things other than light. Then we have the extremes here. We have the archaea. The archaea, their similarities of RNA sequences is why it's closely related to the domain eukarya. In class, I drew this diagram where here you have the domain. You got the eukarya and you got the archaea. Eukarya, and then the true bacteria are the eubacteria. The reason why the archaea are on this end of the domain is because of the similarities of their RNA. They're also going to be the ones that are going to inhabit the extreme environments, whether we have these thermal vents at the depths of the ocean, whether you have these sulfur areas, regions in Yellowstone, or whenever you're flying over, let's say, close bodies of water, such as even the South Bay, landing in San Jose, you can see the coloration. The coloration is not due to rust, but due to the bacteria, the archaea, that are residing in those areas. They have these halophiles, the methogens, and then the extreme thermoacidophiles, just to give you an idea of some of the ways in which they're going to be classified.